Alright guys, this video is going to be the start of chapter 13. We're going to look at the embryonic development of the brain and then different parts of the central nervous system. And so we're going to start off with objective 1. And objective 1 is all about the central nervous system and how it develops. And so just to kind of remind you, we've actually covered this a couple of times, but the nervous system is coming from the ectodermal layer. So remember that early on in development we have 3 layers. The endoderm is going to give rise to the linings of your digestive tract. The mesoderm is the middle layer. That's going to give rise to your muscles and almost all of your connective tissues. And then we have the ectoderm, which is going to give rise to your skin, but it's also going to give rise to your nervous system. And so as the embryo develops, you can just kind of start off as this hollow ball of cells with these different layers. And so the ectoderm is going to specialize and it's going to become, or part of the ectoderm, part of it's going to become skin. But there's going to be a portion that becomes what we call the neuroectoderm. And then it's going to continue to differentiate. And we're going to have an epithelial layer, a covering layer that becomes the neural plate. Then it's going to kind of fold up to form the neural groove. And then what's going to happen are these folds are going to kind of converge and form a hollow tube. And so this right here is really where the beginnings of the nervous system are going to happen, is this neural tube, because this neural tube is what's going to continue to develop and specialize and become the central nervous system. And so in the embryo, the neural tube runs from one end to the other, the anterior to the posterior end. And around 25 days, the anterior end, the front end, is going to become the brain. And then the back end, the posterior end, is going to become the spinal cord. Now the brain is going to be much more complex than the spinal cord, and so we're going to spend most of our video on the brain and how it comes about. And so as the development continues, by the 4th week, we're going to start to see some more complex structures. And so at week 4, what's going to happen, that anterior end, which becomes the brain, is going to form what we call the primary vesicles. They start to enlarge and form these kind of larger chambers that we call the vesicles. And initially we have the prosencephalon, the mesencephalon, and the rhombencephalon. So these are our 3 primary vesicles. Now notice they all have encephalon in them. And encephal refers to brain. Cephal is head. En means inside of, and so encephal means in the head, and it's usually referencing the brain. Now pro means to come before, and so the most forward vesicle, the most anterior vesicle, is going to be called the prosencephalon, and this is going to be what we call the forebrain. The mesencephalon, mes often refers to middle, so the mesencephalon is going to be the one in the middle, and it's going to become the midbrain. And then the rhombencephalon, this actually refers to like a square, so it really doesn't have any relationship to hindbrain, but the rhombencephalon is going to be the 3rd vesicle, and that's going to be the hindbrain. Now, obviously, our brain is going to continue to develop because we have more than just these 3 parts. And so at week 5, about a week later, these vesicles begin to differentiate even further. And so instead of 3 primary vesicles, they're going to form 5 secondary vesicles. Now, the mesencephalon is pretty much not going to differentiate any further, and so the mesencephalon stays the midbrain, which, if you think about the brain as we go through, part of our brain stem is still called the midbrain. But the prosencephalon actually continues to differentiate and forms 2 new vesicles. We have the telencephalon, and that's going to become the largest part of your brain, the cerebrum. And then we have your diencephalon. Now, we actually still use the term diencephalon. So even in the adult brain, the diencephalon is still called the diencephalon. And so we have the diencephalon, which consists of anything related to the thalamus. So we have the thalamus and the hypothalamus. There's also what's called the subthalamus and the epithalamus. And then the rhombencephalon is going to form 2 new vesicles. We have the metencephalon. That's going to become the pons and the cerebellum. And later on, we'll talk about how that these 2 are kind of connected. This is what connects the brain stem to the cerebellum. And so this is kind of going to be a mode of communication between the big cerebrum, or the cerebellum, and the brain stem. And then you have your myelencephalon, which is going to be your medulla oblongata. And so MET also kind of refers to middle, and so pons is going to be the middle of your brain stem. And myel often refers to spinal cord, and your medulla oblongata is going to be the last portion before you get to the actual spinal cord. And so if we look at this breakdown of these vesicles, all of the main parts of the brain are here. We have the biggest part of the brain, the cerebrum. We have the cerebellum, which is the ball at the base of the brain. We have the three parts of the brain stem, which is going to be the midbrain, the pons, and the medulla. And then this diencephalon is actually inside of the cerebrum. So all of our main brain parts are going to be accounted for right here. All right, so this picture kind of breaks it down. In fact, I would encourage you to break it down kind of like it is here, where you have the branches, and then it can branch further. So we have the original embryo with our 3 vesicles. Then those are going to differentiate into the 5 vesicles, and then it goes on to show you what they become. So we have the prosencephalon, telencephalon, cerebrum. Prosencephalon, diencephalon, kind of stays the diencephalon. Then mesencephalon, notice that one stays the same throughout the process. It's still going to be the midbrain. Then rhombencephalon is going to differentiate to those 2 parts, and the myelencephalon is going to differentiate into that. So I would highly encourage you to kind of draw or maybe write it out in this branching type format. All right, here's another way to kind of break it down into all one big picture. So you have your early embryo, your 3 primary vesicles, you have the later embryo, the 5 vesicles, and then you have what those parts form in the adult. Now the spinal cord is going to be much simpler, because if you look at the neural tube, it's essentially a hollow tube. If you look at the spinal cord, there's not really a lot of specialization that has to happen there. It's pretty much going to stay a hollow tube. Now there's going to be some differentiation that takes place, but there's not going to be a lot of big changes like we saw up here in the brain. So pretty much the development of the spinal cord is going to be a little simpler. Now as it continues to develop, we do have cells that make up that neural tube. They're going to proliferate, which means they go through cell division, and they're going to differentiate into specific types of neurons and those support cells, the glial cells. Typically in your spinal cord, your dorsal, which is going to be toward the back of the spinal cord, that's going to be your sensory area. And then the ventral, which is the front of your spinal cord, that's going to be the motor control. And so other than that, there's not a whole lot of parts to the spinal cord like there were the brain. Now we're also going to cover, as we go throughout chapter 13, what can happen when things don't always go like they're supposed to. We're going to look at diseases and disorders as we go through this whole chapter and look at different parts of the brain. And so we're going to look at a couple of neural tube defects. So these actually happen very early on, often before the woman is even aware she's pregnant. So if you looked at what we were talking about earlier, we had development starting as early as, like 4 weeks in, 5 weeks in. And so if those formations are not happening quite like they're supposed to, then it's going to lead to improper formation of your central nervous system. And so this does happen very, very early. And so a couple that we're going to look at is spina bifida, which is a spinal cord defect, and anencephaly, which is a brain defect. So spina bifida literally means split spine, and so this is where when that neural tube is forming, it doesn't completely close like it's supposed to. And so what it can lead to is kind of a hole in the spinal cord, or a hole or missing vertebra along that area. Now it does vary from severity. It's actually fairly common. It's the most common disabling birth defect in the U.S., about 14,000 babies each year. Some of them are easily treatable and fixable, and some of them are very mild, and then others are definitely going to be much more severe. And so we're going to take a look at 3 types. The first one is occulta. This one's fairly mild, and this is where you don't have vertebrae that are formed completely. So if we go back to the picture, you can see that here's occulta. Externally there doesn't really appear to be anything major. There's maybe a hair tuft on the back, but internally you can see that we're just missing a bone there. that the spinal cord is still protected. In fact, a person can probably go almost their whole life and not even realize that they have it. A lot of times they don't know until they have maybe an x-ray or something for some other issue. The next type is meningocele, okay, and this is where the meninges, those protective layers outside of the central nervous system, like the dura mater, arachnoid mater, or pia mater, they actually protrude through the spine and they create this sac on the back of the baby. And so sometimes that malformation is covered by skin and sometimes it's not. But usually this one is pretty easy to fix. They can go in utero and do surgery oftentimes and correct it. But because this really doesn't have nerves, you're not going to necessarily experience major nerve damage or nerve complications. So this one, the symptoms are much less than the myelomeningocele. Now the difference between these two, once again, myelo refers to like spinal cord. This is where a portion of the actual spinal cord or nerves are going to be out in that little sac. And so in this case, when you try to correct it, you will be dealing with nervous tissue, which could lead to more severe complications. They can often close it surgically, but you could experience nerve damage in the process. And so this one is definitely going to be the most severe. And so here's another set of pictures, okay? Occulta, you don't necessarily see any type of major signs. Meningocele, you have the sac. Myelomeningocele, you have the one that has the spinal cord or nerves in it. So definitely going to be harder to fix that one. What could cause spina bifida? A lot of it has to do with a vitamin deficiency called folate or folic acid, which if you've ever had a child or been pregnant, a lot of times you will be advised to take like prenatal vitamins. And one thing that's very prevalent in prenatal vitamins is folic acid. It's going to be important in this early nervous system development. Typically girls and certain ethnic groups are affected more. There are medications or other diseases that could possibly increase your risk of spina bifida. Increased body temp. This is why, like, if you're pregnant, you're not supposed to get in hot tubs or places where it can be extreme heat, because that rise in body temperature could possibly affect fetal development or embryonic development. And then what can happen as a result, like we saw earlier, it really depends on what type it is. But it could be very minimal, or you could have neurological problems. You could have hydrocephalus, which is where fluid builds up into the brain. You could have increased risk of infections or some other later complications like learning disabilities. And then the other neural tube defect that we're going to talk about is anencephaly. So encephal, we said, meant brain. If you put an A or an in front of something, it means not. So it's not that these babies have no brain, but there is a defect where you have either part of their brain or skull missing. Now, unfortunately, this one's pretty severe, and in most cases, babies are probably not going to survive because, number one, they could be lacking brain tissue, and number two, they don't have protection for that brain tissue. And so the exposure of that very delicate nervous tissue is going to often lead to death. And so once again, there could be an environmental factor, there could be nutritional factors like the folic acid, but sometimes it just happens and they don't always know the cause. And that is going to wrap up Objective 1, and I'll see you in Video 2 over parts of the brain.